Hello guys. Now we are going to see first unit in the microbiology. Here we are going to discuss about structure and classification of microbes, morphological types, structure of bacteria, size and forms of bacteria. First, we'll see about microorganisms. Microorganisms are tiny organisms. Microorganisms are tiny. Tiny in the sense small. A small organisms that can only be seen under the microscope. The microorganisms we can't see under our naked eyes. We can see the microorganism by using a microscope because microorganisms are very small. Microorganism found in surround of the air, water, soil, other object and even in the bodies of other organism. We will find these microorganisms everywhere. That is in air, water, soil, other object in the sense non-living things like over the table, over the food particles, etc. And even in the body of other organisms like in the cow, fish, hen, even the human beings. So, the microorganisms will found everywhere. But we cannot able to see our naked eyes. We can found that through the microscope only. Next, moving on to the classification of microbes. The microorganisms are broadly divided into three types: bacteria, protozoa, virus. Bacteria, protozoa and viruses. Again the protozoa will be having a two, that is algae and fungi. So today in this video we are going to see about in detail about bacteria. Bacteria are unicellular. Bacteria are unicellular. Unicellular means uni is nothing but single. Cellular means cell. So bacteria are single cell prokaryotic microorganisms that multiply by binary fusion. Okay, bacteria are unicell prokaryotes. Cells are two types, that is eukaryotes and prokaryotes. So bacteria will comes under the prokaryotes. Example: Staphylococcus, Streptococcus, Neisseria bacillus, Clostridium and Corynebacterium, etc. These organisms can live in soil, air, water and inside the human gut also. The bacterial cell structure. The bacterial cell structure will be having a internal structures, cell envelope and external structures. Internal cell envelope and external structures. The internal structures are cytoplasm, nucleoid, bacterial chromosome, plasmid, ribosomes and storage granules. The cell envelope includes cell membrane, peptidoglycan, cell wall or an outer lipid membrane. This will be found only in the gram-negative cells. Outer lipid membrane will be found only in the gram-negative cells. Next, moving on to the external structures, that is called as appendages and coverings. That is flagella, fimbriae and sexpillus. These are the structure of the bacteria. We'll see now the picture. See, this is a one diagram of bacteria. You can observe. See, this is the cell capsule. Observe the arrow mark. What I am showing. This is the cell wall. Envelope will call. Okay, these three layers will call it as a cell envelope. So, this is the external structure. The internal structure means inside the cell. What are the things are there means? See, cytoplasm is there, DNA is there, ribosomes is there, and plasma membrane, everything will be there. And outer, these are the pillars and flagella. Okay, this is the structure of the bacteria. Usually in the question paper, they will be asking to write the structure of a bacteria. Next, in detail, we will see now. First is cytoplasm. The cytoplasm, or also called as protoplasm of bacteria cell, is where the functions of the cell growth, metabolism and replication are carried out. Okay, cytoplasm is the also called as protoplasm, and the function of the cytoplasm is to for growth of the cell, metabolism and replication will takes place in the cytoplasm only. It is a gel-like matrix composed of water, enzymes, nutrients, waste and gas, and contains a cell structure such as ribosomes, chromosomes and plasmid. You can observe, see, this is completely the cytoplasm only. This is filled with the water, okay, water, air and gases. Okay, so this cytoplasm only will contain these are the ribosomes and DNA and other structures. Okay, it is a gel-like matrix composed of water, enzyme and nutrients as well as waste and gases. Next is moving on to nucleoid. Nucleoid is a region of the cytoplasm where the chromosomal DNA is located. You can observe in this diagram. See, this is completely a cytoplasm, right? So, inside the cytoplasm, this is the nucleoid. Okay, this is the area. The region means area in the cytoplasm where the chromosomes are located. Means DNA is located. Usually in all the other cells, the nucleus will be having a membrane. Means it is the nucleus will be having a membrane covering the nucleus. So, it will give a structure like a round or spherical. But in the bacteria, the nucleus will not be having any membrane. See, most of the bacteria have a single circular chromosomes that is responsible for a replication. Replication is nothing but division. Smaller circular auxiliary DNA strands called as plasmids are also found in the cytoplasm. In this picture, you can observe the nucleus and the chromosome is it is coiled, okay, spiraled. But in few of the bacteria, a single strand DNA will be present, that is called as plasmid. Next, plasmids are a small extra chromosomal genetic structure carried by bacteria. Plasmids are made up of circular piece of DNA. Only the chromosomes has the genetic instructions for initiating and carrying out of the cell division or binary fusion. So, plasmids are the small extra chromosomal genetic structure. In this picture, you can observe this is also a DNA. It is spiral, and here you can observe plasmid. Okay, this is the single strand DNA. Single strand DNA which carries the genetic instruction to carry out the cell division. Next, moving on to the ribosomes. Ribosomes are the microscopic factories. These are the ribosomes. This is a microscopic factories. Like a round dots are there, no? These are the ribosomes, which is found in all the cells, including the bacteria. Ribosome synthesizes the proteins which is needed for the coding of nucleic acid. Okay, ribosomes will synthesize the proteins, that is called as amino acids. Okay, proteins is called as amino acids. So, ribosomes will synthesize the amino acids which is needed for the coding of nucleic acid. Ribosomes are made up of RNA-rich granules. Next, moving on to the cell envelope. The cell envelope is made up of 2 to 3 layers. That is, the interior cytoplasmic membrane, the cell wall and outer capsule. Okay, you can observe in this picture. See here. Here cell wall. This is called as cell wall. Okay, this is called as cell wall, and this is called as capsule, and this is called as plasma membrane. Okay, so the cell envelope consists of the interior cytoplasmic membrane, that is called as plasma membrane. See, don't get confusion. This is completely a cytoplasm. So, just over that one membrane is there, that is called as plasma membrane. That is called as cytoplasmic membrane. And after the plasma membrane, the cell wall is there. After the cell wall, capsule. Okay, 3 layers are there. So, cell envelope is made up of 2 to 3 layers. For few of the bacteria, only 2 layers will be there, and few bacteria, there will be a 3 layer, that is called as outer layer called as capsule. The cell wall, each bacterium is enclosed by a rigid cell wall composed of peptidoglycan molecule. The wall gives the cell its shape and surrounds the cytoplasmic membrane, protecting it from the environment. See, because of the presence of the cell wall only, it gives the shape to the bacteria. And it is made up of what? It is made up of peptidoglycan, okay, molecule. And the cell wall only will give a shape to the cell, which will protect from the environment. Okay, the cell wall only will protect the bacteria from the environment. Next, moving on to the capsule. Some of the bacteria have a third protective covering called as capsule. This capsule is a third protective layer, and the capsule will not be present for all the bacteria. Only some of the bacteria will be having a capsule. Example, E. coli and Streptococcus pneumonia. These are the bacteria which has a capsule. The most important role of this capsule is to keep the bacterium from drying out and protect from a phagocytosis. Phagocytosis is nothing but eating or engulfing. So, this capsule only will protect these bacteria from engulfing. Keep in mind, capsule will not present for all the bacteria. Only some of the bacteria will be having this layer. Okay, this is called as third protective covering of the bacteria. Next, moving on to the flagella. Flagella are the hair-like structures that helps for the locomotion. You can observe in this picture. See, these are the flagellas. Okay, it is a hair-like structure. From the cytoplasm itself, it will start. Okay, from the cytoplasm itself, the flagellas will start, and it comes through the cell walls and it comes outward. That is a hair-like structure which is present over the bacteria, that is called as flagella. And these flagellas are helps for the locomotion. These flagellas can found at either or both the ends of the bacterium or all over its surface. Okay, so these flagellas are there, no? See, this is the flagella, hair like long hair like structures are there, no? This is called as flagella. This can be found either one side, either this side or this side, or both the side, or all over the surface of the bacteria the flagella will be present, and this will help for the motility of the bacteria. Motility means movement of the bacteria. Next, moving on to the pili. Many of the bacteria have a pili, that is called as sex pili. These hair-like appendages on the outside of the cell allow it to stick to the surfaces and transfers the genetic material to another cell. See, these are the pilis. You can observe the difference between the flagella and pilis. You can observe here. See, the pilis are a small hair-like projections, but the flagella is a long one. So, these are called as sex pili or pilis. This will attach us to the surface of the another object or another cells, and it will transfer the genetic material to the other cells. This can contribute to the spread of illness in the human being. So, I hope all of you have understood the structure of the bacteria, right? When they ask you to write the structure of the bacteria, you have to write the diagram. Okay, this diagram you have to write it, and you have to explain about the cytoplasm and nucleus and ribosomes, as well as the cells membrane. Okay, cell membrane. 3 membranes are there. That cell membranes you need to explain, and the flagella you have to explain, and pilus you have to explain. Pili or pilus. Okay, for these things you need to explain it. Got it? Okay. Next, moving on to the classification of bacteria. Bacteria is broadly classified based on the shape, based on the staining, based on the motility, based on the oxygen requirement, based on the flagella. Okay, bacteria are broadly classified based on these, that is based on the shape of the bacteria, based on the staining of the bacteria, based on the motility of the bacteria, based on the oxygen requirement to the bacteria, and based on the flagella. We will see one by one. Based on the shape. Okay, based on the shape of the bacteria are broadly divided into 3 types. That is spherical shaped, rod shaped bacteria and spiral bacteria. Spherical shape. Spherical is nothing but oval or spherical in shape. You can observe here. Just observe the arrow mark where I am showing. This is the oval shape or spherical in shape, called as cocci. Okay, spherical shaped bacteria we will call it as a cocci. Example, streptococcus. Okay, see, this is the coccus we will call. A single one we have called as a coccus, and 2 cocci are there. We will call it as a diplococci. A cluster of cocci are there, that is called as staphylococcus, and see, the cocci are arranged like a chain, that is called as streptococci. Okay, these are the spherical shaped bacteria, we will call it as a coccus. For single bacteria, we will call it as a coccus, and 2 bacteria, spherical shaped bacteria, we will call it as a diplococci. A cluster of bacteria, coccus, are called as staphylococcus. A chain of cocci we will call it as a streptococci. Next, rod shaped. These are known as bacillus. Okay, spherical shaped we will call it as a cocci, right? So, a rod shaped bacteria we will call it as a bacillus. Some rod shaped bacteria are curved, called as vibrio. Okay, see, you can see, observe the curved bacteria, bacillus, we will call it as a vibrio. Example, Bacillus anthracus. See, bacillus will be rod shaped bacteria. Next, spiral. These are known as spirella. If there is a coil is very tight, they are known as spirochetes. Leptospirosis example. You can observe here, these are the spiral bacteria. Got it? Based on the shape, there are three types of bacteria are there. That is spherical shaped we will call it as a cocci, a rod shaped we will call it as a bacilli, and a spiral shaped we will call it as a spirali. Next, moving on to the based on the staining. Okay, here in the laboratory, to identify the structure of the bacilli, we will do the staining. So based upon the staining, again the bacteria is divided into three types. That is gram staining, acid fast staining, Albert staining. So gram staining, there are two types, that is gram positive bacteria, gram negative bacteria. Again in gram positive, cocci and bacillus will be there. The cocci like streptococci, staphylococcus. The bacillus like Bacillus clostridium and etc. Again the gram negative bacteria are the cocci like N. gonorrhea. N means Neisseria. Neisseria gonorrhea and N. meningitidis, etc. The bacillus like Brucella. Next, acid fast staining, that is Mycobacterium tuberculosis, Mycobacterium leprae. Then Albert staining, Corynebacterium diphtheriae. Okay, this staining is nothing but laboratory staining. Okay, laboratory staining. We can observe the bacterial structure and bacterial characteristic by using a staining in the laboratory. So based upon the staining, the bacteria are divided into gram staining, acid fast staining and Albert staining. In the gram staining, both gram positive and gram negative staining will be there. Next, moving on to the based on the motility. Motility means movement. So the bacteria will be two types. The bacteria which will move, we will call it as motile bacteria. The bacteria which are not moving, we will call it as a non-motile bacteria. See here, as I told you in the structure, flagella is responsible for the motility of bacteria. So when there is a flagella only, the bacteria can move. If there is no flagella, then the bacteria will be non-motile. The bacteria cannot be moved. So example for motile bacteria are Salmonella, Vibrio cholerae and Pseudomonas, etc. Example for non-motile bacteria are Staphylococcus, Shigella, etc. Next, moving on to the based on the oxygen requirement. Okay, based on the oxygen requirement. Oxygen requirement means nothing but for the growth and development of the bacteria. For the growth and development and for the respiration, few bacteria requires the oxygen and few bacteria do not require the oxygen. So based upon the oxygen requirement, again the bacteria are divided into the aerobic bacteria and anaerobic bacteria. Aerobic bacteria are the one which requires the oxygen for the growth and development. Example, P. adenias and E. coli, etc. These are the bacteria's names. Don't get confusion. Okay, etc. Again, the aerobic bacteria will be having a two, that is obligate aerobic and facilitative aerobic. Obligate aerobic means these are the bacteria which is needed compulsorily the presence of oxygen. In the presence of oxygen only, these bacteria can be survived. If there is no oxygen, then these bacteria cannot be survived. That is called as obligate aerobic. Next, facilitative aerobics. These are the bacteria which will behave like both aerobically and anaerobically. Means these bacteria also requires oxygen, but a little amount of oxygen is enough for the growth and development and for the survivalence. Next, moving on to the anaerobic bacteria. The anaerobic bacteria are the bacteria which do not require oxygen for the growth and development. Example, Clostridium tetani, etc. Next, based on the flagella. Based on the presence of the flagella, the bacteria are divided into, see, your trichus. Trichus is nothing but flagella. Okay. So, monotrichus. Mono means what? Single. So, only one flagella is there. You can observe in this picture, this is one of the bacteria cell where only one flagella is there. That is called as monotrichus. Example, Vibrio cholerae. Next, lophotrichus. Lophotrichus means a group of flagellas which are present at one side of the bacteria. You can observe at one side of the bacteria a group of flagellas are there. That is called as lophotrichus. Example, Bratunella bacilliformis. Next is amphitrichus. Amphitrichus is nothing but both the side of the bacteria a bunch of flagellas will present. You can observe the differences. See, here only one side the flagellas are there, but here both the side the flagellas are there. That is called as amphitrichus. Example, Spirillum serpens. Next is peritrichus. Peri means all over the surface of the bacteria the flagellas are present. All over the body. That is called as peritrichus. Example, E. coli. Along with that, absence of the flagella also is there. Absence of the flagella is also is there. That is Atrichus bacteria. Atrichus bacteria means there is no flagella. So, if there is no flagella, the bacteria cannot be moved. So far we have seen about the classification of microorganisms. That is, what is the classification? Bacteria, parasite and viruses. Next, after the classification of the microorganism, we have seen about the bacteria in detail. That is, structure of the bacteria and classification of the bacteria. So, by this, the first unit of the microbiology has been completed. Regarding the virus and the other parasites, we will discuss in the another units. I hope all of you have been understood the topic. Thanking you.